Okay, so today we take a whirlwind tour, as I always say, of the cerebellum. So we do the cerebellum in one day. Now, you probably have heard of the cerebellum, and you might have thought it's kind of down there. It's like you don't have to worry about it too much. It's just doing some sort of low level motor thing of some kind. But it's important to sort of keep, the overall picture in mind. So 3/4 of all the neurons in your brain are in the cerebellum. 3/4 of all neurons in cerebellum. So it just on numbers alone, there's a whole lot of them there. Now, the other thing is when you look across the evolution of the cerebellum in primates, so evolution of the cerebellum in primates, what you see is that the cerebellum has actually expanded more than the neocortex in human evolution. So if you look at a monkey brain, we have a much bigger neocortex than a monkey brain, but our cerebellum is way bigger. It's expanded like 2 to 3 times as much relative to the neocortex. So that's a very large cerebellum is a very human thing. It's probably responsible for a lot of human abilities. And then the other thing about the cerebellum is you often think, well, cerebellum is just kind of refining motor stuff of some kind. But it turns out the cerebellum gets input from all different kinds of, cognitive areas. So there's we'll talk about the cognitive cerebellum that gets input from parietal cortex and prefrontal cortex. But it turns out the cerebellum is evolved in emotion. So damage to the cerebellum results in a very characteristic sort of disturbance of emotional processing. So the cerebellum it's maybe not what you thought. So I've started working on the cerebellum for a couple of years, trying to unfold it. And it turns out it has almost as much surface area as the neocortex when you unfold it. So that's the pep talk for the cerebellum. So let's start off. We'll start off with the connections, and then talk about the peculiar structure of the cerebellum, because it's quite different than the neocortex. And then we'll talk about a well-worked out set of experiments on classical conditioning. Now, you might say, classical conditioning, what is that? Is that, that doesn't sound very modern. But we'll see that the basic idea of classical conditioning is to essentially take some arbitrary signal and associate it with an output, very much like an arbitrary symbol. So we'll come back to that. Okay, so let's start off with the cerebellum. So if you put the pons midbrain junction here, so the cerebellum is behind the pons midbrain junction. So here's midbrain and pons. So how does input get to the cerebellum? So there's a ton of input to the cerebellum. We'll just start with the spinal input to the cerebellum. So if you look at, the cerebellum is this huge thing. I'm going to draw it like a frog cerebellum, just like a little strip going across. So that's the cerebellum there. So there's the midline, and the fourth ventricle. I'll just dot in the fourth ventricle like this. So if we look at the main, so remember back in the day we talked about the gracile nucleus. That was the one that did the foot, and then the cuneate nucleus, which is a little bigger, that did the hand. So this is, gracile means light-bodied. That's the one that went up to ventrobasal. And then the cuneate, that does the hand. That one is over here. But if you were doing brain sections, when I was, back in the day when I was teaching anatomy with brain sections, you'd look at the brainstem and you'd struggle to find the cuneate, little cuneate, and maybe grass out a little bigger. And then the students would always ask, well, what's that big thing off to the side? So there's a big thing. It's like twice as big as the cuneate off to the side over here, and that's actually going to the cerebellum. And that thing is called the external cuneate. External cuneate. So it's bigger than the regular cuneate, and it actually is the representation. It delivers basically information from the hand to the cortex. So I didn't put the outside of the brain here. So here's the outside of the brain. Yeah. So that's information about the hand coming in there. So that would be, hand. So there's a ganglion cell going off to the hand, receptor in the skin, comes in to the external cuneate, and that goes up to the cerebellum. Goes up to the same side, cerebellum, because the cerebellum is behind the pons midbrain junction, and the external cuneate is also. So the thing about the cerebellum is the foot representation is slightly bizarre, and I don't know if anybody's come up with a reason for this, but if you look at the foot and the sort of lower body, there's actually some cells sitting in the white matter of the spinal cord. So where is this in the actual spinal cord? So if you draw the spinal cord, there's the ventricle like that, and you have the dorsal horn, and the ventral horn where the motor neurons are, and there's the other dorsal horn, and something like that. So where are these neurons here? They're kind of sitting off in the white matter right about here, just like a little cluster of neurons, and that's called Clark's column. And it's the representation of the foot. So this thing is it goes there. So that's where the foot information gets in. So the foot. So there's the foot, hand. So there's a ganglion cell. It goes out somewhere, maybe into a muscle. That comes in, synapses on the cells in Clark's column, and then those cells go up to the cerebellum. And then we have the face, of course. And so the face is pretty much, just made it, pretty much the same structure that we already talked about, which was that big nucleus up here called the principal sensory nucleus of the trigeminal. So this guy is the principal sensory nuke of nerve 5, the trigeminal. So that's the one that one goes up to ventral basal nucleus to form the face representation up in the cortex, but it also goes to the cerebellum. So here's a ganglion cell come in, it comes into that nucleus, and it goes up to the cerebellum. So that's basically the connections, the spinal connections. And so when you actually look at the cerebellum, you can see that the connections coming into the cerebellum are actually bigger than the connections coming into the cortex. It's like twice as big. It's a huge volume of information going up into the cerebellum. And because of that, that's one of the reasons why people initially thought, well, it seems like it's doing, it's just doing some dirty, down and dirty motor stuff and not doing any sort of cognitively interesting things. But we'll see there's a lot of other connections. And so in the middle here, let's talk about some of the other connections. And there's two types of connections into the cerebellum. So one of them is called mossy fibers. So, because they look ever so vaguely like moss. If you stain them and look at them through the microscope, they look like little globby, sort of mossy things. Mossy fibers. And so an example of mossy fibers are, all these guys here. So all these guys here coming in, those guys are mossy fibers coming into the cerebellum. But there's other mossy fibers coming to the cerebellum, and one big source of input is vestibular input. So the cerebellum really has a lot of vestibular input. So the semicircular canal. So here's the semicircular canals. So you got the semicircular canals, and so there's a hair cell. Ganglion cell. So it turns out the ganglion cells, some of the ganglion cells just go straight to the cerebellum, don't even bother stopping at the vestibular nuclei. So they just connect directly vestibular ganglion. But other ones, go to the vestibular nuclei. And then those have a big connection into the cerebellum too. So there's a huge vestibular input to the cerebellum. And it's involved in sort of coordinating, it's known to be involved in coordinating eye movements. But there's also this huge input from the pontine nuclei. And I mentioned this was the quote-unquote new pathway, new in the last 100 million years, that essentially attaches up the cortex to the cerebellum. And a lot of the cortex goes to the cerebellum, not all of it. So when we talked about the striatum, the entire cortex goes to the striatum. So it turns out not all of the cortex goes to the cerebellum. Infratemporal cortex doesn't go to the cerebellum, but a lot of the rest of it does. And what does this, look like? I've drawn it a couple times. Like if you look at, the neural tube kind of opened up like that. I drew the picture like, you start off with you kind of open it up and you have the sensory part up here and the motor part down here. But if you look in the midbrain, or, in the pontine, where the pontine nuclei are, there's this gigantic thing sort of sticking, like in a human, because the cerebellum has expanded so much, there's this enormous thing, way bigger than the whole brainstem, and that's the pontine nuclei. So these are the pontine nuclei. They act, despite their name, like they're from the midbrain. So if you look at the connections, the connections, come from the cortex, but other things, they come from the midbrain too. And there's also ascending projections, but we'll just put those here. And those guys synapse onto the same side. So the midline, here's the midline, so the cortical input goes down and synapses on the same side of the pontine nuclei on the same side. And then the pontine nuclei send a big connection across to the cerebellum. So that's an enormous input to the cerebellum. And then there's other nuclei that you haven't heard of, but let's just put them down. Don't have to remember, but there's the lateral reticular nucleus. That sounds lateral. It just sounds so generic to me. Lateral reticular, because it's kind of in the reticular formation, but it's kind of off to the side. So where is that guy? Well, if you look at our opened up brainstem, so we've opened up the fourth ventricle. The lateral reticular nucleus is sitting. You have to go back pretty far. So if you go back into the medulla, you see this very large nucleus there, and that guy projects up to the cerebellum on the same side. So this will go, up to the cerebellum. and so left will go to left. And then there's another one called the arcuate nucleus. Arcuate nucleus. That one's up a little bit higher, higher in the brain. And then there's yet another one called NRTP, which stands for nucleus reticularis tegmenti pontis. Tegmenti pontis, which just means the reticular, sort of net-like looking nucleus in the floor of the pons. And so that's another one. That one's a little bit further north, a little bit more toward the midbrain. So that one, it says the fourth ventricle is starting to close. And that's also heavily involved in eye movements. It's down about in the same place as the lateral reticular nucleus. So there's lots of inputs, and if you look at what the inputs of these guys are, there's a whole bunch of there's cortical inputs going to these things. So like just the lateral reticular, for instance, it's got cortex coming down to lateral reticular. It's another way of getting into the cerebellum. It's got some spinal inputs. I didn't draw them here, but they're there. Some spinal inputs. It gets input from, the red nucleus. That's a motor structure, but it sort of goes there. It gets input from the vestibular system. I didn't draw those up here, but that's going on too. So there's a ton of, and I won't draw out all the inputs to these guys, but these guys, there's a big input from the spirocolliculus down here. So there's a lot of wires. Can't remember them all. The whole point here is just that you've got this enormous spinal input, you've got this enormous cortical input coming from the pontine nuclei, and then you've got all these other filtered cortical inputs coming from the lateral reticular nucleus. So the cerebellum is really heavily sort of interconnected with different parts of the brain. So all of these things are mossy fibers. That's why I put them under this. So all of these guys are all mossy fibers. So that's the mossy fibers, and then we have the climbing fibers. So the climbing fibers are a different kind of a synapse. So where do the mossy fibers go? What they do is they come in, they form this little mossy-like thing, and they synapse on the granule cells. And the granule cells have an axon that comes up, and then it splits into two wires like that. And so this is the mossy fiber right here as it comes in, as the information comes in from all these different structures here. So the climbing fibers are different. They actually synapse on the Purkinje cells. We haven't talked about them yet, but I'll just draw a Purkinje cell here like this. So this is like this weird little fan-like cell. It's like a two-dimensional cell. So that's a Purkinje cell. And the climbing fibers come in to the Purkinje cell, and they make lots of synapses, like 50,000 synapses. It's like the strongest synaptic connection in the entire brain, where this climbing fiber will come in and synapse on a Purkinje cell. And where are they coming from? They're coming from the inferior olive. So another olive. Doesn't have anything to do with the superior olive. So if here's our picture of our opened fourth ventricle, if you look down in the brainstem, you see this big thing sticking out. And it kind of looks like, it looks like another little cortex. So it's a folded up structure, the inferior olive, because it's got a fold because it's essentially preserving a map of different parts of the cerebellum that it's projecting to. So it's like a little mini cortex kind of jammed into the brainstem. And so so this guy is called the principal inferior olive. But of course, it wouldn't be complete unless there was some other ones. So there's the dorsal accessory olive and the medial accessory olive. So there's actually there's multiple olives. So remember the superior olive, that was the thing that was like, that had two parts, but the medial superior olive was the thing that was like nucleus laminaris. It's doing the time comparison in the auditory system. So the only thing about, so why was it superior? It's just further toward the front. And so if you just look at a human brain, you'll see two little bumps. One of them is the superior olive and the other one was the inferior olive. So completely unrelated. But basically these guys project up to the cerebellum, but they come from the rhombic lip. So same thing that makes the cerebellum. So, origin from rhombic lip. That's those cells that sort of near the pons midbrain junction that form the cerebellum. But, they act like midbrain. And if you actually look at the, migration pattern, they start at the rhombic lip and they go way back into the brainstem. And so their connection to the cerebellum is crossed. So it breaks the rule of Serino because it's behind the pons midbrain junction. And so they actually have a crossed, in the inferior, all these olives have a crossed projection into the cerebellum. So it's kind of, they act like they were actually in the midbrain, and since they have to go down to the cerebellum, they cross. And when they get to the cerebellum, they form those things. Now, those little climbing fibers that wrap around and make 30,000 synapses onto one Purkinje cell. So now what we want to do is start to zoom in a little bit. And we'll start sort of at a relatively large scale and look at the cerebellum after we've unfolded it. So the thing that's different about the cerebellum, completely different than the cortex. So if you look at the cortex, there's folds in the cortex. Some of them sort of are relatively linear folds, like the superior temporal sulcus or the central sulcus. But the folds go, in all different directions in the cortex. It's not particular, there's no single axis of folding. And the cerebellum is different. The cerebellum has one axis of folding. And it's the same throughout the cerebellum. So if you inflate the cortex, the cortex is folded up. If you inflate it, you sort of get something that sort of looks more or less like a ball. The cerebellum is different. It's just crinkled in one axis. And so if you unfold the cerebellum, it's like you took a piece of paper and just crinkled it horizontally. So it's all squished up into a little blob like that. But if you took your human cerebellum and stretched it out like that and got rid of all the folds, it wouldn't expand at all this way. It's about 10 centimeters. It's about like that. It's about 10 centimeters wide, but it's over a meter, about a meter long. So it's this giant long thing that's all crumpled up just in one axis. And it's very hard to unfold. I spent a year or so trying to unfold it. We finally got it unfolded. Big long thing. And if you look at the overall structure of it, it's kind of sort of a strange structure. There's something called the anterior lobe, like that, and then there's a part in the middle. Let's make it a little smaller. Part in the middle called the vermis, which means the worm. And then that part sort of sticks down a little bit, like that. And then there are these two big long hemispheres that go way down like that. Something like that. So it's got a, so if you unfold the thing, it's quite a strange little man. So this thing is the flocculus down there. So that's the unfolded cerebellum. It's about how big it is if you actually unfolded it. It's this long thing. If you go in the MRI center, you can see up on the wall, there's a nice poster showing it unfolded. So the folds all go this way. There's thousands of folds all just that way. So, now we've sort of unfolded them. So that's a strange structure. So what are the parts? So this guy is called the vermis, which means the worm, and it extends sort of down there. And then these guys are called the hemispheres. So these are the hemispheres. They get really big in humans because that's where a lot of the cortical input goes. And then there's a sort of a separate part down here called the flocculus. So that's particularly involved in eye movements. So that's the flocculus down here. So this is still hemisphere. Still hemisphere. So that's the basic layout of the cerebellum. Now, what do these climbing fibers look like when they come into the cerebellum? So this is the direction. The fold direction is the direction of these parallel fibers. So these guys are called parallel fibers. I'll make another drawing of these parallel fibers. And so the parallel fibers are going in this direction. Parallel fibers. But if you look at the input from the Purkinje cell fans, those little flat Purkinje cell fans are actually perpendicular to the parallel fibers. I'll draw that in a second. But if we look at these climbing fibers, what they do is they come in to the cerebellum and let me draw that in a different color. They form little sagittal projections into the cerebellum. And so what does sagittal mean? So sagittal means like between left and right, the plane that's between left and right. That's the sagittal plane. And so these guys come in and they come in and they form like a little cluster of parallel fibers that are running perpendicular, a cluster of climbing fibers that are running perpendicular to the parallel fibers. And they'll go to several different places in the cerebellum and form these little sagittal zones. So these guys are called sagittal zones. And a number of years ago, somebody found that they were just sort of messing around with stains in the brain, and so they found this one stain. And when you use this stain, what would happen is, especially up in this anterior lobe, is you see these really clear, super clear stripes. And so they were called zebrin, so these are in Britain, zebrin. So zebrin zones. And I mean, it's if you look at the cerebellum, you can't sort of, it looks relatively uniform. And then you stain it with this zebrin protein, and you see zebrin protein, and you see these like absolutely razor sharp little stripes in the cerebellum that are lined up with the way that the climbing fibers come in and form these sagittal zones. So yeah, so that's the strange cerebellum. It's very anisotropic. It's different, it's different in different axes. The cortex isn't like that. If you look at a cell in the cortex, it will typically go out and sort of project in all different directions to its neighbors. Here, it's very anisotropic. You've got these absolutely flat Purkinje cells that are sort of sitting like this, and you got the parallel fibers going perpendicular to them, and you got the sagittal zones. So yeah, it's quite a different thing. So if we zoom in on that, I'll erase this whole thing here and just look, zoom in on the structure of the cerebellum. Now, the cerebellum is a little bit unlike the cortex in that it's got two different levels of folding. The cortex actually has that too. So if you look at like something like the superior temporal sulcus or the central sulcus, like if you go to the MRI center and look at I printed out my brain and tacked it to the front door. If you look at it, you see it kind of looks, it doesn't look, it's a little hard to find like the superior temporal sulcus. And why is that? Well, because it's got a bunch of smaller crinkles in it. And so when we look at the neocortex, it's got two levels of folding in it also. So it's got, it has like the superior temporal sulcus, but if you look at the superior temporal sulcus close up, it's got all these little wiggles in it that are much smaller than the superior temporal sulcus. This is more extreme in the cerebellum. And so if you look at the cerebellum, what it looks like is it has these things called lobules. So as we go sort of from front to back, there's like a chunk that might be one lobule, and there's another chunk. So this would be a lobule. And here it's still folded up. So this thing is called a lobule. And that's the higher level of the larger set of folds. But if you look at the smaller set of folds, those are called folia. And so the smaller set of folds, so you can see that there's a whole bunch of these little things. I mean, I think later on I'll show you a picture of it. So there's like a zillion of these little folia in there. So these guys are called, they're much smaller. So these guys are called, so like that thing is called a folium, one folium. It just means like a leaf. Actually, that's what the word means. So, and the whole cerebellum is completely covered with those. Those are these things here, the things I've drawn here. So these are the parallel fibers are running that way, but the folia are running that way too. So pretty much the whole cerebellum is like that. There's thousands of them, and you can just pull it out like a crinkled piece of paper. So what is the actual structure inside if we just sort of draw one folium. So here's just one folium like that. Okay, so this is one folium, of which there are thousands. So here's one folium going this way. So what does the structure look like? So it's got three layers to it, three main layers. So there's a big layer in the middle that has the Purkinje cells in it. And so what are the Purkinje cells? Well, they're these cells that, it's the most amazing cell in the brain in a way. When Kahala was first looking at these, there are these giant cells. They got 1 million branches on them, and they look like a fan. and they're completely flat. So if you look at them from the surface, they just form like a little thin fan going across the cerebellum like that. They're completely flat, really flat, completely unlike the cortex. So you look at a pyramidal cell in the cortex, and the dendrites go out in all different directions. Here they're just like completely flat. And then you've got the little granule cells. That's the most common cell in the brain. 75% of all the cells in your brain are granule cells. And these guys come up, and then they come out this way. They're going parallel to the folium, and then they go this way down to other Purkinje cells. So somewhere down the line, there's another Purkinje cell, over here sitting in the. And so they're going to run into those other Purkinje cell fans perpendicular to them. So here's the mossy fibers coming up, and there's a little mossy fiber, so those guys are called granule cells. So these are granule cells because they're really small, little granules. And there are some inhibitory cells in there too. There's stellate cells and large basket cells and Golgi cells, not Golgi tendon organs. So this is, let me put that in red. So you got granule cells, those are the most common ones. So this layer is just completely stuffed with cells. So, granule cells. And then these are stellate, these are inhibitory. Stellate, basket, Golgi. So these guys are inhibitory ones. Inhib. And then you got the climbing fibers. So those guys are coming up separately, and they're kind of skipping the granule cell layer. They go through the granule cell layer, and then they wrap around one of these Purkinje cells, make 50,000 synapses. So that's so this is mossy here, and this guy is climbing. Okay, so how does the output get out of the cerebellum? So the output out of the cerebellum comes out of the Purkinje cells. And the thing that really distinguishes this is that it's an inhibitory output. So it's a minus output. And we'll see the same thing in the striatum. So it's an inhibitory output, and that inhibitory output projects to three main nuclei, and so that are in the white matter of the cerebellum. And they have funny names. So this one's called the vestigial. That's the medial one. This one is ridiculously called the globose and emboliform. You don't have to remember that. Emboliform, that means sort of the blood clot shaped nucleus. That's a great name for it. Globose and emboliform. And then this one is called the dentate nucleus. And that's the lateral one. So this is the intermediate. We'll talk about the intermediate one in a second. This guy, sometimes it's called the lateral. So those guys are getting an inhibitory projection from the granule cells, and then they go out, these cells go out, and they go out to things like the red nucleus. So that's an example. And so if you stimulate the red nucleus, you get a movement. If you this is a plus here. And if you stimulate these so these things all together are called the deep cerebellar nuclei. because they're kind of in the cerebellum. They're in the cerebellum, inside the white matter. Here's like white matter of the cerebellum. And so why is that one called the dentate? Well, because it looks like teeth. And so it's full, there's a whole nother little cortex inside there. It's all folded up like that because it's got a map of all the different parts of the cerebellum. It's the dentate nucleus. Now don't mix that up with the dentate gyrus, because that's in the hippocampus. That's also folded up, looks like it has teeth. That's what dentate means. So that's the dentate nucleus. So basically you can get out to the red nucleus, and then this goes up to the thalamus. So there's projections up to the thalamus. So that's how the cerebellum gets out. But unlike the cortex, the main output is this inhibitory one. So if you think about it, in order to make something happen, you have to inhibit the inhibitory Purkinje cells. So less activity in the Purkinje cells allows a movement. And so if you actually go and record in the cerebellum is like rocking. It's just like firing all the time, stopping stuff. Like, don't do that, don't do that, don't do that. And every once in a while it just goes, stops for a little second, then you get something coming out. So that's the cerebellum. So now what about, all this input coming in, visual input, body input, all these different parts of the body, how does it actually work? Well, if you look at one of these folia, so if I just blow up one of these folia, so here, I'll draw it again over here. So here's one of those folia. When people went and recorded from these, well, first when they looked at this, they said, well, there's godzillion numbers of these parallel fibers, and so there must be like some kind of activation running down the cerebellum. And so I put an electrode in here, stimulate, and you'll cause spikes to go backwards through the parallel fibers, and you'll activate Purkinje cells all over the place. But it turns out if you actually just go in there and map, what you find is this insane patchwork quilt. Now, remember we said that there were little discontinuities in the somatosensory cortex? The cerebellum has just gone completely bonkers. And so what happens is you'll get like a little patch of the lip, so a little representation of just like of the map and just a little part of the lip, and then that will be right next to a patch of stomach. And then maybe there's like, a few teeth here, like, 2 teeth, if you're doing it in a cat. And then there'll be like, like a finger, just one finger. And, maybe a little bit of the chin. So it's just like this absolutely insane little patch of little maps, just little maps like about maybe something like about that big. And then it's all like pasted down onto the cerebellum. So it's almost like a computer scientist would say it's like a hash table. It's like you've essentially related all the different parts of the body, because if you're eating something, well, maybe your index finger is going to touch your lip, something like that. Now, we don't know if that kind of structure was discovered in the granule cell layer. So if you want to look at these experiments, they were done by Georgia Schamb and Wally Welker. A long time ago, 1978 was the original experiment. Now, when they did these experiments, they're so kind of like whacked conceptually that people just ignored it. It's still there, it hasn't changed. That structure is still there. But people just are like, it just can't compute, and so it didn't get much attention. But there are these little fractured, and so they call this fractured somatotopy. We already said that somatotopy was fractured, but this is seriously fractured. And there's 2 big maps in the cerebellum. There's one up here, and then there's another big one that's down here. So these are like, second fractured map. This is first fractured map. Now, we don't really know whether this occurs in other modalities. It probably does, like the visual or the auditory system. But it's likely that it does. It hasn't been really completely peeled apart. But so it's just this weird thing. It's got 3/4 of the cells in the brain. It's all folded up in this one axis. So why didn't they sort of believe this? And the problem was that you look at all these parallel fibers and you say like, well, it must be sort of doing something along the axis of the folia. And so how come you can end up with a map? Well, people said, well, maybe there's a map in the granule cell layer. So this guy's called a molecular layer. It has mainly fibers in it. This is the P cell, and this is the granule cell. At first they thought, well, it doesn't have. It probably is irrelevant because the Purkinje cell is getting all these other inputs. But it turns out that if you look at the Purkinje cells, not just the granule cells, there's maps in them too. And why is that? Well, it's because something comes in, hits a granule cell, and the granule cell goes up, and then it comes out like that at you, and then it goes that way. And if you just count the number of synapses, on the way up, it makes maybe 7 synapses on the Purkinje cell, and then on the way out, it makes like just 1. So the projection to the Purkinje cells that are right above the granule cell are stronger. And so if you record from the Purkinje cells, they look just like the granule cells. They have little receptive fields, and they have little fractured maps in them. Okay, so do we have any idea how this works? So what I want to do now is go over some well-studied involvement of the cerebellum in classical conditioning. And when you first hear about this, classical conditioning, you think, wow, that's just not cognitive, that's just like Pavlov and his dogs, conditioning things. But if you think about it, what is classical conditioning? Classical conditioning is basically setting up a symbol, an arbitrary symbol. Because if we look at what classical conditioning is you associate some random thing like a tone with something like a reward, or in this case, an eye blink. And normally a tone is not going to make your eye blink. But if you do classical conditioning, you can essentially wire up some random thing, random tone, and attach it up to something else. So it's kind of an arbitrary connection, like a symbol, like think of a word. What is a word? A word is a sound. Like I say cow, and cow means, if I say cow to you, you get an image of a cow. So somehow you've attached up some random sound, cow, which is say cow to a cat, it's not going to get an image of a cat, of a cow. And so it's like that. That's what classical conditioning is. It's a very simple version of it. And so what was it done with? It was done with a rabbit. And so a number of rabbits were harmed in the process of this experiment, but not during the actual. So here's a rabbit. So if you put a little rabbit, there's a nice little rabbit. And you put a little rabbit in a box, and don't do anything painful to it. And then just put a tiny little air tube like this, and do a tiny puff. Not an unpleasant puff, just a little tiny puff of air. What will happen? The rabbit will just blink its eye. And if you're not poking the rabbit, it'll just sort of sit there, and you can just do a puff again and blink its eye. And so that's what we're trying to study as a very simple, arbitrary symbol operation. And so we have the unconditioned stimulus, which is the air puff. And then we have the conditioned stimulus, which is a tone. So normally an air puff will cause a eye blink. So that's just hardwired. So if we do, the unconditioned response, it's an eye blink. But that's a hardwired one. So, like the unconditioned stimulus just does the unconditioned response. And what we want to do is wire up a tone to the eye blink. So that's the conditioned response. It's also an eye blink. But somehow we want to sort of make an arbitrary connection between this tone and make it go over and then essentially trigger that same eye blink. So so how do you do this experiment? You just pair a tone and the air puff a bunch of times, and then sooner or later, relatively soon, you just play the tone alone, and the rabbit will blink its eye. And so the question is, where is this symbolic bond stored? It turns out it's in the cerebellum. And they weren't looking in the cerebellum. So this is another very good example. I said, when you get an answer that's not what you were expecting, very important data. So they went all over the place. Many rabbits were harmed. Take out the cortex, take out the hippocampus, take out this, take out that. Finally, they took out the cerebellum, and the conditioned response went away. And they said, cerebellum? Well, it can't be in the cerebellum. And so they did a bunch of other experiments. It's in the cerebellum. So how does it work? So let's start off. We've got 1, 2, 3, 4, 5, 6, 7, 9 things, not too many things. So here's the midline. And here's the unconditioned stimulus. So the unconditioned stimulus is the cornea. And the cornea is basically represented in our friend, the principal sensory nucleus of the trigeminal. We've done that a bunch of times. Okay, so that's it represents your face, but it also represents the surface of your eyeball. And so that comes in, and then that projects to the abducens nucleus. Now, we heard of the abducens. That was, we're thinking of that as having something to do with the eyeball. So this is the abducens nucleus. But it also does eye blinks. So that thing goes up to the abducens, and then that comes out and projects to the eye muscle. And so that's both our unconditioned response, but also our conditioned response. So it's both of those. So this is eye blink. And there's multiple pathways. There's some other, the principal sensory nucleus goes to the reticular formation, and that goes, to the eye blink muscles. But that's the hardwired thing. So you've just got this hardwired. It's not a symbol. It's just like, if somebody puffs your eyeball, you blink your eye. So now the problem is, how do we sort of attach some random other junk? Could be anything, like a flash of light, a particular colored leaf, some random tone in the laboratory. How do we sort of connect that up to this, to make a conditioned response? And that's where the cerebellum comes in. So first, how does the conditioned stimulus get in there? So the conditioned stimulus, so that's the tone. So that's coming in through the cochlear nuclei. So there was the posterior ventral cochlear nuclei. We'll call it the ventral cochlear nuclei. Those are we had 2 of them in owls and mammals, but we'll just do one. So that comes in there. And so where does it go? It goes to the pontine nuclei. So the pontine nuclei are kind of, remember, they're sort of they're kind of like even though they're called pontine, they're in the midbrain effectively. And so the projection from the ventricular nucleus to the pontine nuclei is crossed. And then that gets up into the cerebellum. And so here's just one folia. And here's a Purkinje cell. There's a Purkinje cell. And so that's gonna basically get input from a granule cell. So here's a granule cell. Remember, I'll draw that in a different color. So there's a granule cell, little cell, and it goes up like that, and then it sort of comes through, comes out at you, and then it goes in like that. So the pontine nuclei comes in as, we saw that connection in one of our connections as a mossy fiber, and hits the granule cell. Okay, and then how does, we're going to have to sort of like somehow get information in the cerebellum. If the cerebellum is the thing that's storing this mapping between the tone and the eye blink, it's going to have to get information about the eye blink. And so how does that information get in there? So that information comes in through the dorsal accessory olive. So it's not even the main olive, okay? It's the dorsal accessory olive. So here's the dorsal accessory olive, DAO. And so that guy is going to get input from the eye blink information. So that's another connection I didn't tell you about. You know, the principal sensory nucleus projects to the dorsal accessory olive. Dorsal accessory olive. It's crossed because I told you that the inferior olive is this weird thing. It acts like it's in the midbrain also. And so that thing goes up, and that thing goes up and actually forms climbing fibers, because that's what the dorsal accessory olive does. Well, we still haven't sort of wired the whole thing up to the eye blink. We have to get to the eye blink, and so that's the last thing, which is the Purkinje cell comes out and synapses on one of the deep cerebellar nuclei. So here's deep, intermediate, cerebellar nuclei. So that's down here. So this guy synapses on it. And then that thing goes to the red nucleus. That's the last thing, final insult. So it goes to the red nucleus. So this guy is the red nucleus. And we said that was the original motor cortex. So this is the red nucleus. And since that can move the body, how does it move the body? Well, it projects, because it's in the midbrain. That one actually is in the midbrain. It just projects down to the abducens. So that's how so that's where the conditioned response is stored. So the tone comes in, it goes up to the pontine nucleus, pontine nuclei goes into the cerebellum, goes into the deep cerebellar nucleus, and then it goes across to the red nucleus, and then it hits the abducens nucleus. And so that's, so the actual symbol mapping is stored in the cerebellum. And like I said, it was completely unexpected that it should be there. So when they first did this experiment, they found that if you lesion the cerebellum, it would, early in the process, it would make the conditioned response go away. But after the conditioned response is actually, so initially they just took out the whole cerebellum and the deep cerebellar nuclei, but then they figured out it's just the deep cerebellar nuclei. And so now the question is, how do they get, so the pontine nucleus also projects to the deep cerebellar nuclei. And the dorsal accessory olive also projects to the deep cerebellar nuclei. So it turns out after you've learned it, it's actually stored in the deep cerebellar nuclei. Again, like blowing circuits, like could this actually sort of be where things are stored? And so in the last negative 11 minutes, we'll just talk about what is the evidence. I've drawn these things and said this is where it's stored, but what is the actual evidence? So if we go to a small part of intermediate deep cerebellar nuke to show that it's only just a small part of it. So you could, you just have to, it's like 1% of the intermediate deep cerebellar nucleus is where the information is stored. And so you can lesion it, and if you lesion it, what does it do? So the unconditioned response is okay, so you still get an eye blink, so you haven't messed up the eye blink, but the conditioned response just disappears immediately off. So that was the original. After they trimmed down their lesions, they could just make a tiny little lesion there, and you could still condition the animal to do something else, like say, respond with an eye blink to a flash, but that you had interrupted just this particular tone. And then you could also stimulate there, and if you stimulate there, you can show that you'll get you'll get an eye blink after conditioning, not before conditioning. Eye blink, after conditioning. You'll get an eye blink no matter what. Yeah, because that thing is just projecting to the red nucleus, and it can cause an eye blink. It's a map of an eye blink. Yeah, so you'll get the eye blink no matter what, even if the animal hasn't been conditioned. Okay, so that's the evidence that this is the place where the conditioned response is actually stored. But what about the conditioned stimulus? So this is a conditioned response. So what about the evidence that the conditioned stimulus is actually coming through the ventral cochlear nuclei? So in the case of the conditioned stimulus, if we stimulate the pontine nuclei, if we lesion the pontine nuclei first, what happens? So we get no conditioned response to tone, but other conditioned response okay. So we haven't messed up like a visual conditioned response. We just messed up the tone one. And so we know that part of the pontine nuclei that's receiving input from the cochlear nuclei is the thing that's carrying the conditioned stimulus, this arbitrary tone that we just are going to wire up to our eye blink. And then if we stimulate there, you can use as a conditioned stimulus. So what does that mean? That means like instead of playing a tone, you could just do a little stimulation there while you did your air puff. So if you just pair a stimulation and the air puff, after a while, you will be able to just stimulate there and get an actual eye blink. So you can replace the conditioned stimulus, which is a tone, with a tiny little stimulation of a small part of the pontine nuclei. This is just a small, teeny little part that's getting input from the auditory. And then finally, what about the unconditioned stimulus? What's the evidence that the unconditioned stimulus is coming through the inferior olive? And so this was the dorsal accessory olive. So again, you could lesion this, and what happens is the conditioned response goes to extinction. It doesn't disappear instantly, to extinction. So what does that mean? That's kind of an ancient terminology for classical conditioning. If you condition the rabbit by pairing pairing an air puff with a tone. After a while, you play the tone, the rabbit blinks its eyes. But if you keep playing the tone a bunch of times without the air puff there, eventually it says, hey, I don't care, I'm not going to blink anymore. And so that's called extinction of the conditioned response. And in order to keep the conditioned response there, you've got to occasionally do an air puff in order to keep the mapping. Otherwise, it's going to say like, well, that was just some weird thing and I'm going to forget about it. So that's what will happen if you lesion this part of the dorsal accessory olive. It will go to extinction and then you won't be able to train it again. Even though you so it goes to extinction, even with training. So you continue training with the air puff and it's still there for a while and then it disappears slowly. And so that's pretty good evidence that this thing is actually relaying information to the cerebellum about the air puff so that it can be paired with the tone somewhere in the cerebellum. And then finally, you can also use this as unconditioned stimulus. So you can just do a little stimulation of a small part of the dorsal accessory olive, and that will actually serve as an air puff. And so you can pair a little stimulation here with a tone, and then eventually just get a blink to a tone. So this was like years and years of experiments to try to figure this out. Now, so what's the bottom line in the last negative 17 minutes? So the bottom line is what you have here is a general system for attaching anything to anything. The cerebellum is getting tons of, the inferior olive and the pontine nuclei are getting information from the visual system, auditory system, somatosensory system. Like you're playing piano, you're reading notes on the staff. That's a visual piece of information. That could get into the cerebellum and sort of like wire it up to a particular finger. If you're learning words, that's an auditory thing coming in to the cerebellum. And the cerebellum may somehow be involved in actually sort of helping call up the concept of a cow when you hear the word cow. Because, la vache is cow in French, that doesn't work for most Americans, so it's clearly something that has to be learned. So cerebellum is like a general purpose symbol processor to sort of attach up arbitrary symbols or arbitrary sensory things out in the environment with different actions. And not just actions, because the cerebellum, like I said, gets input from all these higher level parts of the cortex, and it projects not just to the parts of the thalamus that go to the motor cortex, but it projects to the parts of the thalamus that go to prefrontal cortex and orbital frontal cortex. And so it's heavily involved not just in direct movements, but in planning for movements and sort of thinking about movements and all different kinds of things. So that's a stretch. So nobody has studied the actual circuitry of the cerebellum in piano playing or the actual circuitry of the cerebellum in word recognition, but we know that the cerebellum is involved. You get activity in the cerebellum all the time when you do any kind of language experiment. If you do a language fMRI experiment, there's activity in the cerebellum. A lot of people just initially ignored it, but no, it's definitely there. Okay, I went ridiculously far over, so I'll stop the recording there and ask for some questions. David?